Welcome to another class of Biology Brief channel. Today we are going to study about the checkerboard of genetic code in easy method. Students may feel very difficult for answering questions from codon or genetic code. They usually make mistakes. We are going to study an easy method to understand the checkerboard of genetic code so that the questions can be answered very easily. Let us move. Checkerboard of genetic code. In the checkerboard, the nitrogen bases are written in the order UCAG. First, you have to by heart this order UCAG. In the checkerboard, there are four rows and four columns. Totally 16 boxes are there. 16 boxes. 4 into 4, 16 boxes. Each box, in each box, there are 4 codons. After all, 16 into 4, 64 codons are represented in the checkerboard. Here, for our convenience, we are assigning a 3-digit number for each codon based on their relative position in the checkerboard. By analyzing the 3-digit number, we can decode the codon by visualizing the group where the amino acids are placed. This is our method. Let us take the columns one by one. Here, this is column number 1. Here, column is vertical one, rows are horizontal ones. Here, in this column number 1, this is row number 1, row number 1, row number 2, row number 3, and row number 4. In each row, the amino acids vary. In first row, the nitrogen base is U. Second row, the nitrogen base is C. Third row, it is A, and fourth row, it is G. In the order UCAG. You have to remember every time UCAG. UCAG in 4 rows. Being column number 1, the nitrogen base is U. U is seen in all these rows. The second is 1. Serial number 1. That means column number. First number is a row number. Second number is a column number. Here 1, 1, 1, 2, 1, 3, 1, and 4, 1. And here U, U, U. 2 nitrogen bases are there, 2 digits are there. Then the third digit is a serial number 1, 2, 3, 4. Serial number in each box. The third digit is the third digit of the number of each codon is the serial number inside each box 1, 2, 3, and 4. Here 1 U, 2 C, 3 A, and G, UCAG. First codon will be UUU. The number will be 111. Second codon UUC. The number will be 112. UUU. You all know that codon for phenylalanine. Phenylalanine. UUU and UUC code for phenylalanine. Now we are going to fill up the amino acids. Then you first relate the codon with the 3-digit number, and by visualizing this column, get to the amino acid. That is our method. Here, look at this. There is a peculiarity in the case of amino acids. 5 amino acids: methionine, phenylalanine, isoleucine, valine, and leucine. 5 amino acids. Methionine has only 1 codon. Phenyl, 2 codons. Isoleucine, only amino acid having 3 codons. Valine, 4 codons. Leucine, 6 codons. This all type of amino acids are present in column number 1. First, we have written as phenylalanine. Then here AUU, AUC, AUA, AUG. That is 314 AUG. We all know that AUG is the start codon which codes for methionine. In this box, 3 positions are vacant. We have to fill up that vacancy with isoleucine. Here in this column, Fourth amino acid is methionine. The other three amino acids are isoleucine, AUU, AUC, and AUA, code for isoleucine. The lowermost column, here four codons are here, U, C, A, G. Four codons are there, all for valine, codes for valine. The four codons, this space will be filled with valine. Here, the remaining is two codons here and four codons here. After all, six codons. Six codons are for leucine. You have to write leucine here and leucine here. First column is completed. Very easy. You have to visualize this one very easily. First, start from UUU. We know that UUU and UUC are for phenylalanine. Then the remaining six, leucine. Then AUG for methionine. The remaining three codons for isoleucine. The last four codons are for valine. 411, 412, 413, and 414 are for valine. First column is over. Let us move to second column. Second column, this is the first row, second row, third row, fourth row. First row, U, C, A, G is the first nitrogen base. Being column number 2, the second number will be 2. Then here, the nitrogen base is C, UC, CC, AZ, and GC. Then, remember this sentence. Sir, proceed through Alatur. Sir, the spelling is different. Sir, proceed through Alatur. S-E-R, right here, S-E-R. Serene, proceed, PRO. Prolin, through THR, threonine, and Alatur for alanine. Second column is completed. Four amino acids are there. Each amino acid has four codons in this column. Serine, prolin, threonine, and alanine. Here we know that CCU, CCC, CCA, CCG. CCC, a homogeneous code. We know that, like UUU for phenylalanine, CCC is for prolin. We have to remember that also. This is about column number 2, very easy. Then move to column number 3. Just difficult this one. We can study easily. Just listen. Here, 8 columns are here. 8 columns are here because each item has 2 codons. So there are 8 columns. Here, first group, row number 1, row number 1, row number 2, row number 3, row number 4. Here being the first nitrogen base U. Here C. Here A. Here it is G. Being third column, the second digit number is 3. 1, 3, 1, 3, 2, 3, 2, 3, 3, 3, 3, 3, 4, 3, and 4, 3. Here, being column number 3, the second digits are 3, and the second nitrogen base will be A. UA, CA, AA, GA. Here, UAU, UAC, UAA, UAG. We know that UAA and UAG are stop codons. There are 3 stop codons, 2 are here. UAA and UAG stop codons. If we imagine an incident when a car comes through a curve and by seeing something, the driver applied a sudden brake, the car tumbles down. When the car toppled down, what happens? The tire will be on top and the driver himself on below, lower side. Unfortunate incident. We are taking this incident for our remembrance only. Tire is on top. Tire is on top and driver himself is bottom. Just write tire here and his here, himself, his. Above the stop codon in column number 3, tyrosine is there. Below stop codon, histidine is there. UAU, UAC code for tyrosine. CAU and CAC code for histidine. Then AAA, AAU, AAC, AAA and AAG. You know that AAA, another homogeneous codon, AAA, code for lysine. So, put lysine in this column. Lysine has two codons, AA and AAG, lysine here. Below lysine, you just analyze this one. GA, we know that gibberellic acid, a acidic plant hormone gibberellic acid. So, we remember here we have to write acidic amino acids. GA for gibberellic acid, acidic plant hormone. So, we add here acidic amino acids. Which are the acidic amino acids? Glutamic acid and aspartic acid. GAU, GAC, GAA and GAG, aspartic acid and glutamic acid. Just above lysine, here lysine, just above lysine, you have to place amides, two amides there. More nitrogen containing amino acids, amides there. Which are the amides? Here, glutamine and asparagine. Glutamine and asparagine. So, column number 3 is completed. Just to visualize. Just slightly tough, but you can easily remember this. First, you start from stop codon. Above stop codon, tyrosine. Below stop codon, histidine. Then lysine. You remember A for lysine. Put the lysine that column. Just below lysine, acidic amino acid GA. Remember GA, acidic amino acid, aspartic acid and glutamic acid. Above lysine, two amides, glutamine and asparagine. Column number 3 is over. Then the last column, that is column number 4. Very easy. Here, first row, second row, third row, fourth row. We put 1, 2, 3 and 4. Here, first nitrogen base is U. Here it is C. Here it is C, A, G, UCAG. Then being fourth column, next digit is 4 for everywhere. 4. Fourth nitrogen base is guanine, G. UG, CG, AG and GG. You put second nitrogen base there. Here, UGU, A, CG, U, A, CG, U, A, CG. UCAG, the order is UCAG, order is UCAG, order UCAG. Fourth, third nitrogen base is in the order UCAG. Here the number is 141, 142, 143, 144. Here 441, 442, 443 and 444. These are number. You have to fill up all the numbers. Here UGA, that is 143. 143, digit number 143 for UGA. UGA is a stop codon. The third stop codon is UGA. Below, just below the stop codon, UGG, 144. UGG for tryptophan, TRP, tryptophan. tryptophan. We know that methionine and tryptophan, both amino acids have only one codons each. So, you put tryptophan in the one place here. Above stop codon, cysteine, amino acid cysteine, UGU and UGC code for cysteine. Then, here in the second box, you have to write arginine. Then here 444, we know that 444 is GGG, homogeneous codon, code for glycine. Here in the third box, you have to partition it. Lower there is arginine, upper there is serine. Arginine and serine both have 6 codons each. Serine is placed in second column and fourth column. Arginine only in the fourth column but have 6 codons. Here this is the column number 4. Amino acid arrangement of column number 4. You just start from UGA stop codon. UGA just below UGA tryptophan. Above 2 codons for cysteine. Then just below the second, in the second box you have to write arginine. In the fourth box there is glycine. Here arginine below, above serine. This is the order of the amino acids in column number 4. You have to visualize one by one, the columns one by one and the serial numbers one by one. Then this is the overall checkerboard of genetic code. There are some peculiarities. We already said there are 16 boxes. In each box there are 4 codons are there. Then if you examine the homogeneous codons, UUU, CCC, AAA and GGG, they have the serial numbers 111. UUU have the number 111, CCC 222, AAA 333 and GGG 444. These are the 3-digit number and homogeneous codons. UUU 111, CCC 222, AAA 333, GGG 444. What are the first codons in this checkerboard? First codons are UUU, UCU, UAU, UGU. The difference is in the case of the middle nitrogen base, the number column. This is in the first column, second column, third column, fourth column. 111, 121, 131, 144 are there 3-digit numbers. The 3-digit numbers of the first amino acid codons of this checkerboard are 111, 121, 131, 141. What about the last row? Last row there is valine, alanine, glutamic acid and glycine. All are in the row, in the fourth row. So GUG, GCG, GAG, GGG. 1, 2, 3, 4 column. First column, second column, third column, fourth column. But the remaining digits are same. 414, 424, 434, 444 are the 3-digit number. You can relate the 3-digit number and nitrogen base of the codons respect to their positions in the checkerboard and find out the amino acids very easily. Then one asks GAG, GUG. We know that these 2 codons are related to sickle cell anemia, point mutation. Which one codes for valine? Which one is that of valine? You just make them into 3-digit number. GAG is 4 UCAG 434, GAG 434. It is in the third column. GUG 414. We know that the valine is the 4th last amino acid of first column. That is the number is 414 and the codon GUG for valine. GAG is for glutamic acid. You can easily decode any codon, making them, converting them into 3-digit number and assign the respective column and find out the amino acid. Just like we are also going to discuss some questions related to codon or genetic code. Let us see. First question. Choose the correct option of amino acids coded by the following codons. Here 4 codons are given. The codons are homogeneous codons. UUU, CCC, AAGG. You are simply know that UUU codes for phenylalanine, CCC for proline, AA for lysine, GGG for glycine. You can simply know and you can correct answer the question very easily. The fourth option will be the answer. You can also convert them into their respective numbers: 1, 1, 1, 2, 2, 2, 3, 3, 3, 4, 4, 4. You can convert this codon to their 3-digit number, and by visualizing the columns, you can easily find out and reach to their respective amino acids. Very simple. Moving to second question. Match the columns 1 and 2 correctly. UUU serine, UCU cysteine, UAU phenylalanine, UG tyrosine. If we imagine these codons, UUU, UCU, UAU, UGU, just you carefully examine the codon, you can see that all the codons are placed at the first row. Here 1, 2, 3, 4. 1, 1, 1, 1, 2, 1, 1, 3, 1, 1, 4, 1. These are their three-digit numbers, which are the first amino acids in the row. Here phenylalanine, here serine, here tyrosine, here it is cysteine. You can easily decode that one. UUU phenylalanine, UCU serine, UAU tyrosine, UGU cysteine. What is the answer? A3, B1 here only. So our answer will be fourth option. You can easily answer such type of questions. Let us move to third question. Which one of the following pairs of codons is correctly matched with their function? Pair of codons are given and their function. They are stop or starting or coding any amino acid. Just analyze this. UAG, you know that is stop codon. UGU is not a stop codon. It is the last amino acid present in the first row. UGU, first row. It is code for cysteine. So this option is a wrong one. AUG and ACG. ACG is not a start codon, or ACG will not code for methionine. So this option is also a wrong one. Last option, just look last option. UUU, we know that code for phenylalanine and not code for leucine. That option is also correct. Three options are wrong. The answer will be third one. Let us GCG and GCU. Let us find out the three-digit number. 4 UCAG, 424 UCAG, 421, 424 and 421. These two are present in the second column. Second column, fourth row. Second column, fourth row is nothing but alanine is the amino acid, and this option is very well. The third option is very, very correct, and it is our answer. You can confirm that by this three-digit conversion. What about the fourth question? Match. Select the incorrect match. You have to select the incorrect match. Remember that word, incorrect match. Yes, here you just analyze these four options. Second option and fourth option, both are ACC. We know that codons are amino acid specific. That means each codon codes for only one amino acid. ACC, either code threonine or serine. Option 2 or 4 will be our answer. We have to find out which is our answer. You decode this one. ACC, UCAG 3, 2, 2. UCAG 3, 2, 2 is our three-digit number. Then second column and third row. Second column, third row. What about second column? Sir, proceed through Latur is our code. Threonine. Second column, threonine. Threonine, third row. Threonine. ACG, threonine is correct. So our answer will be fourth option. ACC, not code for serine is our answer. Very comfortably you can reach the answer. Then our last question. Which are the following code for amino acids proline, valine respectively? Respectively, listen that word. proline and valine. Sometimes we reach the answer from codon to amino acid. In this question, you have to travel from amino acid to codon to find out the correct answer. Let us see proline. First one is proline. We know that proline is present in the second column. Valine is present in the first column. We have to remember or visualize. Proline in the second column, valine in the first column. What about second column? Second column we already know. In second column, Sir proceed through Alatur. Second column, proline is present in the second row. Second row, second column, second row. What about second row? Second row, UCAGC. CC. CC will be first two codons. CC. Here CC, you can find CC here. CC. Then what about first column? First column, we know that valine is the last amino acid. Valine. In the fourth row, first column, fourth row valine. Fourth row means G is the first nitrogen base. Being first column, U will be the second nitrogen base, GU. So you can easily find out fourth option is our answer because proline starts with CC, valine starts with GU. Fourth option is our answer. I thought that this would be very useful for you in answering such type of questions. Thank you very much.